Okay, today I'm going to give a brief overview of viruses. We're going to talk about what is a virus and virology and their genetic material. The study of viruses is called virology. And what is a virus? A virus is, in fact, a genetic element that can replicate inside of a living host cell. Now, inside the cell, that's important. They cannot, in fact, survive outside of the host. They are an obligate intracellular parasite. They can infect both prokaryotes and eukaryotes. And one of the big things is they're not cells. A virus is not, in fact, a cell. Um, so they do contain their own nucleic acid genome that allows for replication and travel from cell to cell. This is called a viron. So a viron consists of a protein shell called a capsid, and inside this capsid is where the genome is contained. Okay, so most bacterial viruses are naked with no further layers. Here's an example of a naked virus. So if you notice here, they have the capsid. It's composed of the capsomeres, and inside is the genome, the nucleic acid. Versus a naked virus, you have an enveloped virus. The difference here is they have an extra layer. They have your nucleic acid, your capsid, and then this envelope surrounding it. And if you notice here that within an enveloped virus, the capsid and nucleic acid, this is called a nucleocapsid. The viron protects the viral genome when the virus is outside the host cell, and the proteins on that viron surface are important in attaching it to its host cell. The viron also contains one or more virus-specific enzymes that play a role during infection and replication. Okay, so once this viron gets inside the cell, it can do a couple of different things. So the virus can replicate and destroy the host in a virulent or lytic infection. So in this type of infection, the virus redirects the host metabolism to support the virus replication and assembly of new virons. Eventually, these virons are released, and the process can repeat itself onto new host cells. On the other side, some viruses can undergo a lysogenic infection. In this type of infection, the host cell is not destroyed, but it is genetically altered because the viral genome becomes part of the host genome. Okay, so let's now talk about that genome. So we know all cells contain double-stranded DNA genomes, but a virus's genome consists of either DNA or RNA, and then can be subdivided based on whether the genome is single-stranded or double-stranded. There are a few unique types that use both DNA and RNA. So the viral genomes can be either linear or circular, and single-stranded viral genomes may be of either plus sense or minus sense. Genomes of the plus sense have the exact same base sequence of that of the viral mRNA and will translate to form viral proteins. And those of the minus configuration are complementary in base sequence to the viral mRNA. And so viral genomes are usually typically smaller than those of cells as well. You can classify viruses based on the types of hosts they infect, as well as their genome structure. So we have many different types. We have bacterial viruses, archaeoviruses, animal viruses, plant viruses, protozoan viruses, and a couple more. Bacterioviruses are called a bacteriophage, sometimes referred to as just a phage. Okay, so now that we know a little bit about viruses and their genome, how they're classified, we're going to move on to the life cycle of a virus. Now, for a virus to replicate, it must induce a living host cell to synthesize all these essential components needed to make the new virons. It can't do it on its own. It has to have the host, and the host has to be alive. Dead host cells will not replicate viruses. So a cell that supports the complete replication cycle of a virus is said to be permissive for that virus. In a permissive host, the viral replication cycle can be divided into these 5 steps. So step 1, we have attachment or absorption of the phage viron. Next, we have penetration of the viral nucleic acid. Step 3 is synthesis of viral nucleic acid and protein. Now, it uses all of the host cell's energy and mechanics to do this. Step 4, all of the newly made nucleic acid and protein are packaged. And step 5, they are released. This release happens towards the end of the maturation phase and is a result of either cell lysis or by budding or excretion, depending on the type of virus. The number of virons released per cell, called the burst size, varies with particular viruses and the particular host cell and can range from a few to 1,000. The duration of the virus replication cycle also varies. It can be anywhere from about 20 minutes to 60 minutes in bacterial viruses and 8 to 40 hours in animal viruses. Okay, so in review, a virus is an obligate intercellular parasite that requires a virus is an obligate intracellular parasite that requires a suitable host for replication. A viron is the extracellular form of a virus and contains either an RNA or DNA genome inside of a protein shell. Once inside the cell, either the viron or its nucleic acid redirects host metabolism to support virus replication. The virus replication cycle can be divided into 5 major stages: attachment, penetration, protein and nucleic acid synthesis, assembly and packaging, and viron release. Now, viruses can only replicate inside of their host cells. Remember, viruses are not a cell themselves. They are completely dependent upon their host. I hope you guys learned a little bit from this simple overview of viruses. And Dr. Blair, you have my permission to use this video in the future. Thank you.